Today we will discuss about the bacterial cell in details. The bacterial cell has various components. That is, first of all, the flagella. These are extremely thin hair-like appendages. They come out through the cell wall and originate from basal body, structure just beneath the cell membrane in the cytoplasm. The flagella is the plural, whereas the flagellum is the singular. If there is only one flagellum is present around the bacterial cell, that is known as the flagellum, and if many flagella are present on the outer surfaces of the bacterium, that is known as the flagella. The flagella are very special types of the structures of the bacteria. These are not present on all types of the bacteria, but they are specific for specific conditions or habitat. The flagella are made up of the special type of the protein that is known as the flagellin. Classification of bacteria on the basis of flagella. Based on presence of the flagella, pattern of attachment of flagella, and the number of flagella, bacteria are classified into different taxonomy groups. For example, first of all, the Atrichus. The bacteria that has no flagella, that is known as the Atrichus bacteria. The Monotrichus, the bacteria that has only one flagellum is present around the surface, that is known as the Monotrichus. Third is the Lophotrichus. Flagella is present only at one pole of the bacterium, that is known as the Lophotrichus. Lophotrichus, that it indicates from its terminology. This is not present on specific place, but it depends upon the polarity of the bacteria. The third is the amphitrichous bacteria. The amphitrichous, just like that of this, is a condition when tuft of flagella and each of two poles is present. This is the first pole, this is second pole. In these conditions, the two flagella are present on both poles. Both poles mean positive or negative poles. The fourth is the peritrichous flagella that surround the whole cell body of the bacterium. Most of the bacilli and the spiral-shaped bacteria have the flagella, whereas the cocci very rarely have the flagella. The other structure that is known as the pili and their functions. The pili are hollow, non-helical filaments, appendages, present straight rod-like structures present outside of the bacterium. They are smaller than the flagella and are not involved in the motility. The flagella, functions of the flagella is to move from one place to another place of the bacterium, whereas the pili are also such type of the appendages, but they are smaller and not involved but mobility, but they are generally present in the gram-negative bacteria. The pili are specially made up of the protein that is known as the pilin. They are primarily involved in mating process between cells called the conjugation process. Some pili function as the means of attachment of bacteria to various surfaces. The pili are specially involved or formed when the bacteria has become in such conditions when they are going to reproduce themselves, or they made from one bacterium or one strain to the other strain of the bacterium. In these conditions, both bacteria produces the pilins or the pili, and they made each other. The other structure is the cell envelope. The cell envelope is consisted of the capsule, slime, and the cell wall. First of all, the capsule. Bacteria produce capsule, which is made up of repeating polysaccharide units, and a protein of both capsule is tightly bound to the cell. It has thicker gummy nature that gives sticky characteristics to the colonies of the encapsulated bacteria. It provides pathogenicity. Pathogenicity means that these capsules are attached with the host cells from where bacteria has to produce the diseases. The conditions in which bacteria produces the diseases, that is known as the pathogenicity. The other structure is the slime. Some bacteria are covered with loose soluble shield of the macromolecule, which is called as a slime capsule, and slime provides greater pathogenicity to bacteria and protects them against the phagocytosis. So slime is very elastic in nature, and very soft material that helps the bacterium to attach some other types of the surface host or other bacterium. They protect against the phagocytosis. It means that the chemical nature of the slime is toxic, so that the other type of the chemicals or the bacteria cannot harm the specific bacteria having the slime over the coat. The third is the cell wall. Beneath the extracellular substances and external to cytoplasmic membrane, cell wall is present, which is a radius structure. It determines the shape of the bacterium. Cell wall also protects the cells from the osmotic lysis. Cell wall is only absent in the mycoplasma. The mycoplasma are also special kind of the bacterium that has no cytoplasm cell walls. That is the cell envelope. The cell envelope of the bacterium is of two types according to the discovery of the Christian Grams. The Gram is the scientist who discovered two types of the bacteria on the basis of their staining process. The first of all, the gram-positive bacteria, and then is the gram-negative bacteria. Gram developed a technique of gram stain. Bacteria could be divided into two groups based on their response to gram staining procedure. By this procedure, the gram-positive bacteria are stained purple, whereas the gram-negative bacteria stain in pink. They retain the secondary dye in color. However, such type of the ability to absorb the color or the stains is present in the cell wall of the bacteria. This is a chart describing the characteristics of the gram-positive and the gram-negative bacteria and the comparisons. The number of layers of gram-positive bacteria is 1, whereas gram-negative bacteria, the number of layers is 2. The chemical makeup of gram-positive bacteria is the peptidoglycan, which is 50% of the dry weight in some bacterial cells, teichoic acids and the lipoteichoic acids. This is the chemical nature of the polypeptide glycan. Whereas in case of the gram-negative bacteria, the polysaccharides, lipoproteins, peptidoglycan, and 10% dry weight of the some bacterial cells, this is material is present. In case of the overall thickness, the 1 to 4% lipids, which is 20 to 80 nanometer in thickness of the gram-positive bacteria, whereas the lipids are 11 to 12%, comprising of the 8 to 11 nanometer thickness of the gram-negative bacteria. The outer membrane in gram-positive bacteria is not present, whereas in case of the gram-negative bacteria, the outer membrane is present. The periplasmic space present in some gram-positive bacteria, whereas they present in all bacteria of the gram-negative bacteria. The permeability of different types of the chemicals or the liquids or some other types of the molecules is more permeable in case of the gram-positive bacteria, whereas less permeable in case of the gram-negative bacteria. Then the cell membrane. Just beneath the cell wall is the cell membrane or the plasma membrane, just like that of the other cells of eukaryotic cells, but the chemical composition of the cell membrane of bacterium or prokaryotes is different. It is very thin, flexible, and surrounds the cytoplasm. Plasma membrane is very delicate in nature, and damage to it results in death of the organisms. Because this is very unique and special type of the structures or the covering or the envelope that cover the protoplast of the bacterium. Bacterial membranes differ from eukaryotic membranes in lacking sterols such as the cholesterols. The functions of the cell membrane is to regulate the transports of proteins, nutrients, sugars, electrons, and other metabolites which is required by the bacterial cells. The bacterial uh, plasma membrane also contains enzymes for the respiratory metabolism, whereas this process is not present in the eukaryotic cells. These structures of the gram-negative bacteria originally demonstrated different types of the chemical compositions or the chemicals present, which is the bacterial cell membrane or the plasma membrane, just like that of you can see over here. The lipopolysaccharides, this is outer membrane, this is the cytoplasmic membrane, phospholipid bilayer, peptidoglycan, this is present, which is not present in the eukaryotic cells. This is periplasmic space, which is not present in the eukaryotic cells. The lipoproteins is also present in the eukaryotic cells. The porins, just like that of the channels, they help the transportation sign across the membranes.